Hello and welcome to this episode of CMN Live. I'm absolutely delighted today to be joined by Dr. Cecilia Jimenez-Mallebrera, who is the Director for the Laboratory of Translational Research in Children's Neuromuscular Disorders at Barcelona's San Juan de Dios Institute. Welcome, Cecilia.
Thank you. Thank you very much, Karen, and thank you very much for this invitation.
You're welcome. I'm really looking forward today to hear about the amazing work that you've been doing within developing new therapeutic approaches to neuromuscular diseases. But before we get into that, why don't we start with neuromuscular diseases more generally? What can you tell us about this class of diseases as a group? How do they present themselves? What is their prevalence and sort of what is the current treatment landscape more generally?
Okay, so they are a very heterogeneous group of disorders. So they are mainly inherited disorders, although there are some forms which are acquired. They are monogenic, and they can present at any time in life. They are very heterogeneous genetically and also clinically. So far, there are more than 600 genes that have been identified as a cause of a neuromuscular disease. And there are different subgroups of neuromuscular disorders. So, for example, we have diseases, anything that can impact into the muscle and how the muscle weakness and strength can produce a muscle disease. So, for example, we have diseases that will affect the motor neuron in the spinal cord. We have diseases that affect the axon. And then we have diseases that affect the junction of the axon with the muscle cell, which is an area called neuromuscular junction. And then we have those diseases that affect primarily the muscle fiber. So that's why they are very heterogeneous. And also because skeletal muscle is a highly energy-demanding tissue, there are metabolic disorders like mitochondrial diseases that can also present as a myopathy, as a muscle disease. So that's why they are very heterogeneous. And the individual prevalence of each of them can be low, and generally they are considered rare or ultra-rare diseases. But there are some studies that indicate that if you take all of the neuromuscular disorders as a whole, they can be as prevalent as Parkinson's disease or ALS in Europe. So as a whole, they are a group of quite common diseases. But as I said, they are very heterogeneous.
Yeah. I guess that poses a great challenge for treatment, and I don't think the current treatment landscape is very good. Could you comment perhaps on that? Are there good treatments for any of them that really work?
I mean, traditionally there have been diseases which are very difficult to treat because of this heterogeneity and also because of the target tissue is the muscle. So, of course, the muscle occupies a large percentage of the body, and it's difficult to reach all skeletal muscles. In the last 10 years, that has changed a lot, and in particular in the forms of muscle disease, which are more prevalent. So I'm sure that everyone is mainly familiar with Duchenne muscular dystrophy and spinal muscular atrophy. Those are two of the most common and most widely studied neuromuscular diseases. So they have been pioneered in the sense that the first treatments approved for the neuromuscular disease were for Duchenne muscular dystrophy and spinal muscular atrophy. There were some treatments that alleviate some of the symptoms. So, for example, antifibrotic. They have been used for Duchenne muscular dystrophy for a very long time, but it wasn't until 2017 that the first treatments based on antisense oligonucleotides were approved for Duchenne muscular dystrophy and also for spinal muscular atrophy. And the reason is because those diseases have been widely studied. There has been lots of investment on those diseases and lots of efforts by all the research community. Okay. However, there are many other neuromuscular diseases that are kind of they are taking advantage of everything that we have learned from Duchenne muscular dystrophy and for spinal muscular atrophy. So slowly we are seeing other neuromuscular diseases that have treatments, not treatments, but there is already some advancements at the preclinical level for more several forms of neuromuscular diseases. But most of them, they still lack a curative treatment.
Right. Yeah, so if we were to hone in on what you've been working with, so you've been working with the collagen 6 related muscular dystrophies. So perhaps we could go a little bit more into detail now on those and what kinds of mutations and genes are causing those diseases and what kind of symptom presentation do they have.
Okay, so collagen 6 related disorders, they are actually considered a spectrum of disorders because there are different severities. So on one hand, we have the more severe patients which have what is called Ullrich congenital muscular dystrophy. And then on the other hand, we have milder patients which have the Bethlehem myopathy form. And in the middle, we have intermediate forms. That is because when they were described, they were considered two different diseases. But then when the genetic cause was identified in 2001, it was found out that actually the genetic cause was related to the same group of genes. Okay, so these diseases, they are caused by mutations in any of the 3 genes that encode for collagen 6. Collagen 6 is one of the 28 types of collagens that we have, and is made of 3 different proteins, 3 different peptides. So we have mutations, possibly mutations in the three genes. Most of the mutations, they are dominant. So 3/4 of the mutations that cause collagen 6 related diseases, they are inherited in a dominant way. And that means that we only need to have one copy of the gene which is mutated to present the disease. Sometimes they are inherited, and sometimes these mutations, they are de novo, which means that they appear for the first time in the patient that we are diagnosed or the patient that attends the clinic, but is not inherited from the parents. And in terms of the clinical symptoms, as all muscle diseases, they are characterized by muscle weakness and hypotonia, but in particular, there are other very specific clinical features, which are hyperlaxity. So the patients have, particularly the children, they have very lax distal joints. They also have contractures, which are very limiting for the day-to-day life, and also skeletal deformities. So, for example, they will develop severe scoliosis that requires surgery. And that the reason because that those symptoms appear is because collagen 6 is not only found in muscle, it's also found in the tendons, in the ligaments. So it's like a connective tissue disease.
Right. So much of your work has been focused on understanding the causes of these diseases and mutations involved to identify biomarkers for diagnostics, but also to try and pave the way for new therapies. So yes. I realize we're not there yet, although you've done lots of amazing work, which we'll get into. What would an ideal treatment, in your opinion, look like for the collagen 6 related dystrophies? And how early in life would it actually need to be given for maximal effect?
Yeah, that's a very, very good and very important question. I mean, like any other monogenic disease, ideally what we would want to do is to permanently correct the genetic defect. Yeah. And that is why, although we can use drugs or other things that go against the symptoms, the best thing is always to try to correct the primary genetic defect. And that is why technologies such as gene editing or base editing offer many possibilities. So, for example, in the case of collagen 6, as I said, many of the mutations are dominant, and they can be caused by a change in one single nucleotide. So, for example, base editors, they provide a lot of hope, because if we can correct those single nucleotide mutations permanently, then we should be able to revert the phenotype. Of course, that's still very far because there are many concerns regarding in vivo treatments with gene editing, but correcting the mutations will be ideal. We have been working as well with traditional CRISPR-Cas9 technology to silence the mutated allele, which is a different strategy. It's not so precise. So, for example, because many of the mutations are dominant, if you eliminate that copy of the gene, the one that is mutated, you can prevent the dominant negative effect of those mutations. So it's better to, let's say, it's better to eliminate that copy which is not correct and only have one copy that functions. And that is the basis of the work that we did, which was published a few years ago, in which we used traditional CRISPR-Cas9 technology to produce double-stranded breaks and by means of non-homologous enjoining, to be able to eliminate the mutation, one of the most common dominant mutations in collagen 6 genes. So ideally, we will want to correct the mutations, and now we are working with base editors to try to correct one particular mutation, which is a glycine to arginine substitution, which is quite common in collagen 6 related dystrophy. So doing that, we will be able to permanently correct that at the genomic level, and therefore that protein will form normally and will function normally, expectedly.
That's very obviously very promising. Given the heterogeneity of the diseases and the fact that, they're monogenic, but there are many different mutations involved. Now, I don't know how big the gene is, but how, obviously gene replacement strategies, that's not what you're working on, but they offer the benefit, I suppose, of replacing the entire gene. Whereas the base editing approach will correct individual mutations, but you would probably never be able to capture all patients in that way. So what are the advantages of the permanent gene correction, where you would need to devise individual strategies for individual mutations, versus a gene replacement strategy where you may be able to treat a lot more patients with one treatment?
Yes, there are different approaches, and it depends on the type of mutation. So in collagen 6, there are also mutations which are mutations that could benefit from gene replacement. So there are also homozygous mutations that cause the complete loss of one of the collagen 6 alpha chains, although they are less frequent. But it's also a strategy. So some of the mutations will benefit from gene replacement therapy. It's not something that we are working at the moment, but it's something that other groups are working, because that is also a possibility. But as I said, for the dominant mutations, you cannot replace the gene, because that will not work. What you need to do is to correct the specific mutations. So of course, there are advantages to gene replacement, but depending on the type of mutation, that is not the best strategy. You need to correct the dominant mutations. But there are also homozygous mutations that can be corrected in that way. That is also a possibility. There are many other possibilities. So, for example, there are versions of Cas9 that they will target the mRNA, like Cas13. So that will also be a possibility. Maybe there could be also strategies aimed at the epigenetic correction. For example, if we could find a way of upregulating collagen 6, for example, like is being done for other forms of congenital muscular dystrophy. And there are other types of molecules, like antisense oligonucleotides, that can also be used to silence dominance mutations. But it really depends on the mutation. It's true that there are some mutations that are more frequent in collagen 6. So there is one deep intronic mutation, which accounts for about 20% of the patients with collagen 6 related diseases, and that can be targeted with maybe just one strategy. But it really depends on the mutation that you want to target.
Right. So the toolbox is big, but we probably need all of the tools to solve,
Exactly.
problem.
We are looking, at personalized medicine. Yes.
Sure. So maybe we could go more into now what you've been doing. So what strategy? You mentioned that you were previously using CRISPR-Cas9 gene editing, and now you've moved into base editing. So could you tell us about your strategy? You mentioned you're going after one mutation. How do you deliver the reagents, and how far are you with them, with the experiments actually?
Okay. So first of all, when we are performing what we call proof of concept studies, which is the first phases of preclinical studies, those studies are conducted in cells. So in our group, we have been working, as I said earlier, with CRISPR-Cas9 to target a particular mutation, to provoke double-stranded breaks, and then by means of non-homologous enjoining, we showed that in fibroblasts from patients, so that was our cell model, which is actually the cells that express collagen 6 in some tissues. So we were able to demonstrate in fibroblasts from 5 patients with the same mutation that using that strategy, we were able to eliminate a high proportion of the mutated copies of the COL6A1 gene, and that strategy was able to improve collagen 6 at the protein level and improve the function of the cell. So at the cell level, that was quite successful. And in that case, when we're working with cells, we can use reagents such as lipofectamine or other reagents that we can use to deliver the CRISPR-Cas9 tools. In our case, we started first working with plasmids, but the problem is that when you work with primary cells, they are very difficult to transfect. I mean, they are very difficult for the plasmids to enter the cells. So we use the ribonucleoprotein. So basically the Cas9 protein coupled with the guide RNAs. And that worked very well. And also it has advantages because when it is in the protein form, there is less chances of being genotoxic or of target effects. So in the cells, in vitro, delivering the RNP complex with lipofectamine worked very well. But that is, of course, something you cannot use later on when you go to patients. So there are different strategies to deliver CRISPR-Cas9 components, and one of the main limitations is the size of the Cas9 gene and the cDNA for Cas9 or the Cas9 protein. So most studies so far, they have used AAV vectors or AAV dual systems, so basically carrying the components in 2 vectors and using them to transduce the cells simultaneously. But we have been working with other systems based on lipid nanoparticles. So far, we have worked with a particular type of lipid nanoparticles that has been developed by collaborators in an institution in Barcelona. And what we have shown is that we can conjugate, so we can bind all the components of the CRISPR-Cas9 system to these lipid nanoparticles, which is also something that you need to optimize. I mean, when you start working with these tools, first of all, you need to optimize the condition of this conjugation. And we have shown that using that, we can deliver the CRISPR-Cas9 machinery to the cells. And then once inside the cell, what we need to study is whether that system is effective as well as effective as using, for example, lipofectamine, which is the basic reagent. So at the moment, we are optimizing the conditions using lipid nanoparticles in the cells, and we have also started to do some studies in mice, so to do some in vivo biodistribution studies. But there are different vectors that you could use. So apart from lipid nanoparticles and AAV, you could also use extracellular vesicles, organic nanoparticles, for example, gold nanoparticles, they have been used as vectors for Cas9 protein and Cas9 machinery. So there are different delivery vectors that you can use. The problem is when you go in vivo, because in vitro studies are one thing, but in vivo studies are a different thing.
Yeah. How easy will it be to target the muscles, do you think? And can you learn from previous attempts by others in the field or even by some of the existing treatments that are out there? How do you think you would go about that?
Yes. I mean, as I said, the difficulty with muscle is that you need, in most of these diseases, you need to target all the muscles. Even if in the patients there will be some muscles which are more affected than others, in reality you need to be able to reach all the muscles. In the case of collagen 6, within the muscle, the target cell is not actually the muscle cell itself, but is a cell which produces collagen 6, which is a type of fibroblast which is in between the muscle fibers. So you need to reach the right tissue, but you also need to reach the right cell. So as I said earlier, a large proportion of research is being done with AAV vectors, with natural capsids, meaning existing AAV vectors. The problem they have is that they tend to accumulate in the liver, for example, and they reach muscle because there are already approved treatments for other muscle diseases which use these AAV vectors. But ideally, we want them to be more specific for muscle so that the dose of the vectors that we need to use is smaller and therefore you reduce the toxicity. So in the last few years, there have been some developments improving this type of AAV vectors and making them more tropic for skeletal muscle. So there are actually evolved versions of the AAV vectors, which are called Mayo-AAV or AAV-Mayo, that have been published and that are being used in preclinical studies, which are much better at targeting skeletal muscle. So we can learn from that, and we can use as well those vectors. Regarding lipid nanoparticles, in our experience, there isn't anything yet which is specific for skeletal muscle. And because lipid nanoparticles, there is a huge variety of composition and a huge variety of different types of lipid nanoparticles. So I think at the moment that field is lagging behind in terms of AAV vectors. They offer some advantages in terms of biocompatibility, in terms of safety, in terms of size. The advantage of the lipid nanoparticles is that you are not limited by size. You can bind on the surface or encapsulate large molecules, so that's a big advantage. But there is still no standard formulation for skeletal muscle. You can do different modifications. So, for example, you can try to add molecules to the surface of the lipid nanoparticles that will guide the lipid nanoparticle to skeletal muscle, for example, specific peptides or specific molecules, but it's still a field which is under development.
Okay. Just thinking again about the severity of some of the diseases, in some cases can be quite severe already early in life, and just the timing of giving the treatment, since you cannot reverse the damage once it has been done, and also the challenges that you just outlined in terms of delivery and reaching all of the muscles. Would there be any case for treating very early in life and potentially even at the in utero stage, if one knew that those diseases were present? Where are these diseases included in prenatal screening programs, or when are they typically identified?
For the case of collagen 6 related dystrophy, is not, I mean, because there is no effective treatment yet, and then they are not included in these programs. And as I said, there are so many different neuromuscular diseases that it will be difficult. Of course, the congenital forms, as I said, there are different severities, but the congenital forms, the sooner we could treat them, the better. Mm-hmm. But at the moment, there hasn't even been a clinical trial with advanced therapies for these diseases. So I think that, first of all, we will need to go through the clinical trials and probably treat children which are not as young. And then after that, see, whether they are safe to start with, and then maybe consider treating them as soon as possible. But we are still not in that at that point with collagen 6 related dystrophies. I mean, for example, for spinal muscular atrophy, since there are now treatments that are working in several places in the world, it's been included in the prenatal newborn screening. But that's not the case for collagen 6 related dystrophy yet. I think we still have a long way to go. But of course, if we are talking about a congenital disease, ideally we would want to treat as soon as possible and reverse the disease as soon as possible.
Yeah. So just thinking again about, you mentioned that you had done some preliminary experiments in mouse models. Is it too early to say anything about how that looks yet?
Yes, I mean, our preliminary studies, they are the biodistribution studies, okay. So they are not efficiency studies. So okay. We've gone from the proof of concept studies in cells, where you kind of start with several candidate molecules or, designs, and then you look at, the response, the specificity, potential toxicities, and then we moved into mice, a mouse model, looking at mainly if we can reach, if we provide the machinery, the CRISPR-Cas machinery or the antisense oligonucleotides with the lipid nanoparticles, whether we can reach skeletal muscle, because as I said, there is not much work done with lipid nanoparticles for this type, to carry this type of molecules to skeletal muscle. And so far we can see that they reach skeletal muscle. They also accumulate in the other tissues, like it was expected, like in liver, kidney, and lung, but they do reach skeletal muscle. So we are changing the formulation and trying different functional units, so things that we can attach to the lipid nanoparticles to see if they are more specific for muscle. But we have not done efficiency studies because we need to do those in a mouse model that carries the mutation or the equivalent human mutations that we want to correct. Okay. That's still within a laboratory setting, so it's not kind of regulatory preclinical work, which is a completely different and more complex setting.
Yeah, that was actually one of my next questions. with diseases that are rare, how accessible are the models, or is that also something that you have to develop, the mouse models?
Yes, the mouse models, we have developed a mouse model which is specific for that particular mutation. There are several mouse models for collagen 6 related dystrophy, and the first one was developed many years ago for collagen 6A1 by an Italian researcher. But since we are talking about personalized medicines and targeting specific mutations, we need to have mouse models that have those mutations. So you can either work with a humanized mouse model, which means that it's a mouse model that contains the sequence of that region exactly is the same as the human sequence. So then everything that you do in the human cells, you can do it in the mouse model. And there are humanized models for collagen 6. We took another approach. We developed a mouse model that has the equivalent mutation to the mutation that we want to correct, but is not humanized. Just because developing humanized mouse models also can be quite difficult technically, and at the time we did not have the experience to go into that. So we have developed a mouse model, but it's actually, we have shown that it's quite a good model and it replicates some of the aspects of the pathology. So we can then in the next phases work with that mouse model, and also maybe we can we collaborate with other groups that have the other different mouse models as well, the humanized one. So maybe we can also test our candidates in those other mouse models.
That's great. So you've been busy.
Yes.
Yes. So we're actually almost out of time. So time flies when you're having a good chat. So just sort of looking forward then, obviously, it's a long path to develop a new treatment, especially when you need to develop models and understand the diseases also along the way. So, I'm not going to ask you anything about when you think this will be in the clinic, because I think that's not something that you can answer. And I think it's also unfair to try and guess these kind of things. It's a sensitive area. So instead, I will just finish by asking you what your future plans are with your research and sort of how you plan to navigate this in the next few steps.
So at the moment, as I said, we've been working with base editors, which provide many advantages because they don't do double-stranded breaks, so they are safer and they are more specific. But we want to improve the efficiency of our results with the base editors, optimizing the conditions, first of all, in cells. Then, because we are aware of the concerns regarding the genotoxic effects and also the pre-existing immunity to Cas9, we are collaborating with several groups. On one hand, we have been collaborating with the group of Sangarsai in Memphis. So we have been looking at the genotoxic effects, so the genome-wide effects of our guides, the ones that we use for CRISPR-Cas, to see if they cut anywhere else in the genome and which are the places where they cut in the genome. So that's quite... you have to do quite a detailed study because there are different techniques that you can use and each of them gives you different information. So what we try to do now at the moment is assessing the safety at the genome-wide level of the guides that we are using on one hand. So that's very, I think, very important. Mm-hmm. And then on the other hand, we are looking as well at the pre-existing immunity to Cas9 from SP-Cas9, which is the one that we have been using in children because most of the studies have been done in adults. So we know that in the normal adult population, we have both antibodies and cell pre-existing immunity to Cas9 protein from SP, from Streptococcus pyogenes. So we are doing a similar study, but in children, in the pediatric population. So on one hand, we are doing that. On the other hand, we are trying to improve the specificity of our base editor, the specificity and efficiency of the base editing strategy. And also we are going to continue with the delivery system. So trying to test a wider range of delivery vectors. Probably not just the lipid nanoparticles, because as I said, it's a complicated field, but also trying other types of vectors.
Yeah, excellent. That sounds incredible. I think that brings us to the end, but I do hope that we can stay in touch and maybe you'll come back later on at another time and give us an update.
Thank you. Yes, that would be great. Thank you very much. Yeah,
it's been really great having you here today. It's been really informative. So I just want to say thanks to you, Cecilia, for taking the time to tell us about your work. And thanks to everybody for listening in. In case you missed any of this or would like to rewatch it, you can see it on our YouTube channel and also on LinkedIn and on our website under CMN Life. So thanks, everybody, and take care.